Even here one more point, as the sodium, as I said, it has come 2 times. Again it is recycled back by our sodium potassium pump. As potassium comes inside, the sodium goes for the exchange. So as the sodium is going out, now as the sodium is more in interstitial fluid, this sodium has come into the lumen side. Always water follows sodium, so no need to explain about it. As the sodium is coming, even water comes along with it into the lumen side. So next what happens is, the negative voltage of lumen plus the positive charge sodium ion across the tight junction between the cells. So you can see this is between the cells, the tight junction, it is passing because of negative voltage of the lumen, and it pulls the positive charge. As the lumen is now negatively charged, it pulls the positive, sodium is positively charged, so it pulls the positively charged sodium across the tight junction between the cells. So the overall movement of the sodium bicarbonate, see sodium, so this red mentions that which have come into the lumen, the sodium, the bicarbonate from the blood into the duct. It exerts an osmotic gradient even for the water into the pancreatic duct, forming an isoosmotic bicarbonate solution. So it is called as isoosmotic bicarbonate solution because here sodium, water, bicarbonate, 3 solutions are coming now into the lumen. This is called as isoosmotic bicarbonate solution. So here you can see this is isoosmotic bicarbonate solution. So by this we have understood the process of secretion of pancreatic bicarbonate solution. So now let us see how it is regulated, the regulation of pancreatic secretion. So mainly we have 3 regulators, same like our gastric physiology, like gastric secretions: acetylcholine, cholecystokinin and secretin. So let us see how they will regulate, but here they have some difference. Let us find out. So acetylcholine is it's looking similar. It's activated from our parasympathetic, especially a parasympathetic nerve, that is vagus nerve ending. So from the vagus nerve ending, this acetylcholine is released, and even by some other cholinergic nerves in our enteric nervous system, when it gets stimulated, even from that the acetylcholine gets released. And cholecystokinin is secreted mainly by duodenal and upper jejunal mucosa when food enters. Of course, when the food enters in the small intestine only, all these processes are triggered. Without food, all these are not triggered. And next, coming to secretin. Secretin is an important difference with the gastric secretion and pancreatic secretion we need to observe. So secretin is mainly secreted here by duodenal and jejunal mucosa whenever there is increased acidic food entering the small intestine, or you can say whenever there is acidic chyme entering the upper small intestine, especially duodenum and jejunum, then the secretin gets released. So here you can see the acetylcholine and cholecystokinin both stimulate the acyanide cells, which mainly useful for the production of pancreatic digestive enzyme, but small amount of water, electrolytes also go with the enzymes along with them because of this pull, osmotic pull or any other charges pull. But secretin mainly stimulates the secretion of higher amount of water of sodium bicarbonate by pancreatic ductal epithelium. So if all pancreatic stimuli occur together at once, it leads to higher increase in greater secretion, because you can see if acetylcholine and CCK are activated, mainly the pancreatic enzymes are released, but secretin is activated, is leading, in the name itself you can see it's like secreting. What it is secreting? Mainly in the pancreas, it is useful for higher increase in secretion of bicarbonate solution. But if you are seen in our gastric secretions, the secretin is a negative role, it's playing a negative role, that is secretin is like an inhibitory factor of gastric secretion, if you remember. Why it is inhibiting also we have learned the reason, that as the food is already in the small intestine, we need to inhibit now the secretions in the GI, in the stomach, because already the small intestine is full. So for a while we need to stop the food over stomach. After the small intestine food clears, then we can allow the secretions of GI. So that is the function of secretin difference in a pancreas and a stomach. So next, coming to the phases. So as we already seen in our gastric secretion, we have 3 phases: cephalic phase, gastric phase, intestinal phase. So even the pancreas have the cephalic phase. Here what happens, first the brain signal comes, acetylcholine is released from a nerve endings, especially parasympathetic nerve like vagus nerve endings, and next here the enzymes are released. We already discussed acetylcholine and CCK mainly release the pancreatic enzymes. So here you remember, in the cephalic phase 20% of enzymes are released along with some water, electrolytes. They also go into the intestine. But remember, here the cephalic phase is after the meal. So after the meal 20% of enzymes are released, and in the gastric phase like about 5 to 10 10% of enzymes are here also they are released after the meal in the gastric phase. Next, coming to the intestinal phase. So here the chyme is coming from the stomach, which try to enter the small intestine and increases secretion mainly in response to hormone secretin. So this intestinal phase is dependent on our hormone secretin. So secretin we already discussed, it increases the bicarbonate ion secretion. So this tries to neutralize the stomach acid in the chyme. So actually secretin is a polypeptide which is made up of 27 amino acids, and it's an inactive form is called prosecretin, or also called as S cells, which are present in the mucosa of duodenum and jejunum. So this S cells get activated whenever the food comes, especially in our intestinal phase. So in the intestinal phase, as the secretin is activated, it leads to more secretion of bicarbonate. So when acid chyme of pH which is less than 4.5 to 5 enters the duodenum, it causes the mucosal release and activation of secretin, which is absorbed later into the blood. And this this too potent, means the main activator of this secretin hormone is HCl release, because HCl is the major acid. So because of release this HCl, the secretin gets activated, and this in turn leads to, of course, increased bicarbonate release but decreased chlorine release. This is very important. We have seen there, like here you can see sodium bicarbonate is released more. We have seen even the chlorine is released, but only least amounts, means very low amounts. Mainly the bicarbonate and sodium and water are mainly released. So we also call this solution, already we discussed, isoosmotic bicarbonate solution. So next we'll move on to duodenum. What happens in duodenum? So after this we have saying from beginning, because of secretin this sodium bicarbonate is released. So in duodenum, whenever this acid comes, this reaction takes place. HCl acts upon with sodium bicarbonate. So because of this reaction, it leads to formation of salt and carbonic acid. And again, the process, the carbonic acid in turn divides into carbon dioxide and water, and this carbon dioxide now gets absorbed in blood, or it may be also expired by our lungs. So this is how, whenever we want to alkaline or neutralize the acids which are coming from the stomach into the duodenum, and they prevent the duodenal cancers, which play a very important role in preventing the duodenal cancer. And also peptic digestive activity by gastric juices in duodenum is blocked. This we already discussed in our gastric secretions. So bicarbonate ion secretion by pancreas mainly provide the pH for the action. They also try to provide the pH for the action of enzymes which function optimally in slight alkaline or neutral medium, like of pH 7 to 8, and the pH of sodium bicarbonate is 8 average. So you can also remember the pH of this. And next, coming to CCK. So now let us see how the cholecystokinin tries to control the digestive enzymes which are secreted by pancreas. So presence of food in the upper small intestine cause the release of cholecystokinin, which is a polypeptide with 33 amino acids. So before we already discussed, secretin is having 27 amino acids, whereas cholecystokinin is having 33 amino acids. It is released from I cells, but secretin, as far as it's released from S cells, CCK is released from I cells in the mucosa of duodenum and also in the upper jejunum. So this release of CCK mainly result from the presence of proteases and peptones, products of partial protein digestion and long chain fatty chains. So because of presence of proteins and fatty acids, this CCK is released in chyme coming from the stomach. So secretin is released whenever in the chyme there is more acidity, but this CCK is it's like different process. It is released whenever the chyme is having more protein content and more fatty acids content. So CCK mainly lead to release of enzymes by acyne. So this is very different. You can see secretin is useful for the release of bicarbonate solution from the ductules or ducts, but the CCK is trying to release the enzymes from the acyne, not from the ducts. It is acting on the acyne. So it is same as vagal stimulation for 70 to 80% of total secretion of, we already discussed in which phase this CCK release. 70 to 80% means it is released in, not here, just it is released for 70 to 80% of total secretion of pancreatic digestive enzymes after meal. So after completion of the meal, this try to act on our proteins and fatty acids which are present, but this secretin is different. It acts whenever it comes itself, but this is after the meal. So the total amount of secretion for This is about the pancreatic secretion for each day. The pancreatic secretion for one day is like about 1 liter of pancreatic juice to function, to have this all this kind of function. Well, let us see some textbook diagrams and complete this lesson. So here we have one chart like a summary of pancreatic enzymes. We have proteolytic enzymes, amylatic enzymes and lipolytic enzyme. Let us see what they convert. So these enzymes mainly are the proteolytic enzymes are trypsin, chymotrypsin, carboxy polypeptidases, and amylatic enzymes are pancreatic amylase. Lipolytic enzymes are pancreatic lipase, cholesterol esterase and phospholipase. So first let us see the proteolytic enzyme function. So mainly they are useful for breakdown of protein into peptide. So protein into peptide, this function, and split some peptides into individual amino acids. And next coming to amylatic or pancreatic amylase, it tries to hydrolyze the starch, glycogen and other carbohydrates, remember except cellulose, to disaccharides or trisaccharides. So this amylase convert the glycogen carbohydrates into disaccharides or trisaccharides, and these proteins they break down the proteins into peptides or amino acids. And the lipolytic enzymes they hydrolyze the neutral fat into fatty acids and monoglycerides, or hydrolyze of cholesterol esters in the case of cholesterol esterase, and phospholipase split the fatty acids from phospholipids. So it nothing but it is converting the phospholipids into fatty acids. So these are the important functions of different kinds of enzymes, and this diagram we already discussed. And here we have a comparison of HCl, soap and peptone. So actually this experiment is done on the duodenum part of our GI system. So in duodenum what happens if there is HCl? So you can see whenever there is HCl, there is larger release of water and bicarbonate, sodium bicarbonate from our especially pancreas, we just now studied. Whenever there is higher acid, it tries to neutralize it so that more amount of bicarbonates are formed when compared with enzymes, because there is nothing to digest as more HCl. But now you can understand, if soap comes means fatty acids are coming. So when the fatty acids coming, even here also water and bicarbonate release is somewhat more, like almost like equal. Enzymes are also more, now they need to digest them again. Whereas when the peptone comes, you can see a different scenario here. When the peptone comes, now peptone means here especially are they convert into peptides like that, right? So here more the enzyme should be released when compared with our bicarbonates, because here nothing to neutralize, just we need to now break them down. So according to the substance we eat, according to the chyme, there are the differences of release of either bicarbonate and water or enzymes. You can see clearly by this we can understand. And you can see the regulation of pancreatic secretion, as we discussed. The acid from stomach release secretin from wall of duodenum, fats and amino acids cause release of cholecystokinin, as we discussed, and secretin and cholecystokinin absorbed into bloodstream. The vagal stimulation release enzymes into S-cyanine, and secretin causes copious means higher amount of secretion of pancreatic fluid and bicarbonate, whereas cholecystokinin causes higher secretion of enzymes. So here by this only, here also we can find out which is an action of cholecystokinin and which is an action of secretin. So you can see here as the bicarbonates and water are more, so this is an action of course by secretin, and here you can see in the peptone as here more enzymes, so CCK works more here. So like that you can correlate. So next we have here the secretion of pancreatic enzymes. So everything we have covered, just if you want we can check this diagram to understand how this everything is getting converted. You can see amylase acting on starch, lipase acting on fats, trypsin on protein, how it is converting. So that's it. By this we complete our chapter, exocrine pancreas.